Assalamualaikum guys. Now I'm going to tell you about celiac disease. This is one of the most important diseases in the GIT, and I'll tell you the pathophysiology of this disease and all the signs and symptoms related to it, all the tests we are going to conduct to confirm the diagnosis. So let's just start. I'll be studying this topic from big medium robins, and I have added all the points from the big robins, so it will be easy for you to cram it and have the concepts about celiac disease. Now let's just discuss the topic. Celiac disease. In celiac disease, we eat gluten diet, which includes wheat, rye, barley, and all the stuff which has gluten containing diet. Now, the gluten diet is composed of proteins and starch. Starch is easily digested through the body, while protein enters into the stomach, and from stomach it enters into the small intestine. In normal individuals, gliadin is degenerated it is digested by the IgA antibody. Now what happens is that when gluten enters into the stomach, normally as it is a protein, so there will be proteases, enzymes in the gastric proteases, small intestine proteases, or pancreatic proteases that will digest this protein. Now what happens, gliadin is not digested by all these proteases, so it enters into the lumen of the small intestine. Now how it is degraded in normal individuals? It binds to IgA, which is present normally in the lumen of small intestine attached to the mucosal membranes. So it enters into the it binds with the IgA protein, and this complex is destructed. So in this way, gliadin is removed from the body. What happens in patients of celiac disease? Gliadin enters through the small intestine. Now, gliadin is actually a DMI. Gliadin is actually a protein, D28 amino acid containing protein. That was originated from gluten. So it enters into the lumen of the small intestine, where it binds to the IgA. It forms the complex. It is not degraded or digested. It binds to the transferrin receptor that is normally present on the cells of the small intestine, that is a membrane. So it binds to the transferrin receptor, from with which help it is transcytosed to the enterocytes, which are the normal cells of the small intestine. Okay, it is transcytosed through it. Transcytosed meaning it enters into the lamina propria of the small intestine by passing through the cell. Okay, now it enters into the lamina propria. Once it enters, the gliadin encounters a protein, that is tissue transglutaminase enzyme. Okay, this tissue transglutaminase enzymes removes the amino group. I have written in the theory as well, like this. Now gliadin and IgA binds to the transferrin receptor, and it is transcytosed to the lamina propria. Here it is present, here where tissue transglutaminase enzyme is present. This deamidated gliadin now is eaten up by the macrophage, which is normally lining the cells of the small intestine. As always there are in the whole GIT, you have macrophages which eat up all the debris and extra foreign agents, and is responsible for protection. So this deamidated gliadin is eaten up by the macrophage. Now we know the mechanism behind the macrophage, that they have a major history, that is MHC2 and MHC1 molecules. So MHC is major histocompatibility complex. Like this, you can see the structure. This is a macrophage, and it is MHC2 protein that is presents the deamidated gliadin. and this is represented by this MHC2, which is present on the membrane of macrophages. Now now the T helper cells, that is CD4 cells, recognize this deamidated gliadin, and as it is CD4 positive cells, so they follow this pathway, that is they release inflammatory cytokines. Inflammatory cytokines include interferon gamma and tumor necrosis factor. Now these cytokines are released and they destroy all the epithelial cells of small intestine and destroy the villi. So we have this whole complex of cascade of events that occurs in the celiac disease. Now the villi is lost, there is lymphocytosis as there are abundant T cells, and now next mechanism is once it releases inflammatory cytokines, the other what T cell do is it also stimulates the B cells. Now what is the function of B cells? To produce antibodies. This produce antibodies anti-gliadin, anti-endomysium, TTG, which means tissue transglutaminase. B cells produce antibodies against the gliadin, against the tissue transglutaminase enzyme, and also the endomysium. Now what is endomysium? Why B cell is producing anti-endomysium? Is it is said that the structure of tissue transglutaminase also correlates with the muscle fiber enzyme. So mistakenly it recognizes it as TTG and also starts producing antibodies against the endomysium, which is the inner layer of muscle fiber. Now but it is important for the diagnosis, so so it can also be related. This is the whole mechanism of celiac disease. Now just come to the sign and symptoms of the disease. Now what are the symptoms? As we have, what we eat is not digested, so there occurs abdominal distension, weight loss, diarrhea, anorexia, this kind of. And one thing that is associated and which is very important for celiac disease is dermatitis herpetiformis. What does that mean? It's actually a skin rash. That is a clinical presentation of the disease that occurs in individuals. It is not a definite reason behind the disease, but there could be, we can see patients of dermatitis herpetiformis because it is related to celiac disease. What is the pathology behind this? Is that Ig is normally present in the blood, and mistakenly it binds to the tissue transglutaminase enzyme that is present in the dermal papilla of epidermis. So in epidermis, the IgA binds to the TTG and stimulate the neutrophilic infiltration. Now the neutrophilic infiltration appears as rash. So this red rash on the body skin is known as dermatitis herpetiformis, which is a very important feature of celiac disease. Now this is all about celiac disease, and now come to the tests. I'll teach you with the this is all the diagrams I have shown you. I guess it's all clear now. And now come to this morphology. Now for clinical testing, we usually take the biopsy which is from the duodenum or proximal jejunum, because it is a portion that is highly exposed to the gluten diet. So we take the biopsy of duodenum or proximal jejunum. Now what is evident in the histopathology of celiac disease patient is that we have intraepithelial lymphocytosis, which means, as we have seen that in this diagram, there is cytosed, and in the lamina propria there is the whole complex cascade of events leading to B cell and T cell infiltration. So we have intraepithelial lymphocytosis. Similarly, crypt hyperplasia, which means that in this diagram normally the structure of small intestine is that we have villi and there are shallow crypts. Now in the celiac disease, the crypts are very deep down and it results in crypt hyperplasia. Similarly, there are villi we can see, and in this patient of celiac disease there is loss of villi and there is flattening of the villi. So we have intraepithelial lymphocytosis, crypt hyperplasia, and villus atrophy. So this is all we have in this celiac disease. Some important few points are, we have with the patients of celiac disease, we have high risk of small cell adenocarcinoma. And for tests, so we take a biopsy in which we can see this histopathology, and similarly we have other clues as well, which include some CBC, in which in CBC there is both the dimorphic picture. We can see microcytic and macrocytic anemia because of B12 deficiency and iron deficiency. Okay, so this is all about celiac disease. I hope you liked the video. Don't forget to subscribe my channel.